Sure. So today's talk would be on emergence-led biology breakthroughs and the rise of systems thinking, and specifically to understand how we are able to transform understanding of life and its complexities. So as we begin, I would like to take you through a series of images. Imagine a flock of birds, thousands of them, strong and wheeling and diving in perfect unison. There's no conductor, no script, just an intricate dance of individuals, each responding to its neighbors. This is emergence in action. This is a phenomena called murmuration in birds. The same thing happens when ants come together, form colonies, and create anthills. So we are at the cusp of biology today, where emergence is not just a metaphor, but is actually a guiding principle, just like in the case of the anthill or murmuration. Life, as we see it today, is not a series of isolated components of molecules, of genes, of proteins, but it's actually understood as a complex network. And there's been a paradigm shift that unlocks breakthroughs to revolutionize medicine, to transform our environment, and eventually hope for a healthier and more sustainable future. So let's start off with what would be the outline of my talk today. So I will cover some aspects of systems thinking and emergence, in which I'll talk about what is a system and what is system thinking. So how you define a system eventually ends up in the understanding of systems thinking, and that's what I'm going to walk you through. I'll talk about the systems biology approach, which we use in the lab, and I will talk about in this context integrative biology or modern biology, which combines 5 great ideas that have been existent for the past century or so, including one very interesting one which relates to what Manoj was saying a little earlier about cell phones. It was a concept called omnis cellula e cellula, which was brought out by Virchow, a pathologist from Germany, and he's sort of considered as the father of pathology. And he essentially, what this means is that all cells come from a single cell. So we will talk about how this concept eventually ends up in tackling brain cancer and chemoresistance with respect to what I do in the lab, antimicrobial resistance, and if we have time, a little bit about the cell factories. And hopefully, if we have more time or during the question and answer session, we can discuss how we can move beyond biology and use systems thinking to solve some global problems. So what is a system? So a system is basically more than just a collection of parts. It's a set of interconnected elements that work together to produce specific behaviors or functions. And the key concept here is that the whole is greater than sum of its parts. So what are the key components of a system? The 3 primary components are elements, the interconnections, and the function. The elements are the individual parts. The interconnections define how these parts interact, and the function essentially is the overall purpose or the behavior of the system. Let's dwell a little into the importance of these relationships. So elements are often most visible part of the system, but surprisingly changing them has minimal effect on the system. What is more important and what has most impact is actually changing the relationship between elements. For example, if you change the textbooks, the curriculum, the administrators, the presidents, it doesn't have a major effect on the system. Whereas, if you change the interaction between the people, societies, and, for example, the president or the prime minister, that would have a bigger impact. So that's what I'm trying to get at. So essentially, it is the flow of information through these elements and changing relationships that actually change the behavior. And the interconnections act like the glue between each of these components. So as I mentioned, function is typically the least obvious part of the dynamic system. A system is generally dynamic because it changes with time, over time. And the change in purpose generally changes the system majorly. So what do I mean by that? So Raytheon was an American appliance company, and it was most famous for making parts like motors, vacuum tubes, etc. But today they actually make weapon defense systems. So it was not quite obvious by just looking at the parts that they used to manufacture that they would eventually end up making missiles. So these are some of the examples of complex systems, and I think the ones that all of you are probably most familiar with are the internet, climate, societies, organizations, economies, probably even ecosystems. But if you are not in the life science realm or in the medical field, I would think you would be less familiar with these systems like brains, physiologies, organisms, and lesser so gene networks and chemical networks. And I will touch upon some of these in my conversation today. Now, some of the properties, key properties of complex systems are that they are adaptive, that is, they change with the environment. So if the environment changes, they change. They're quite unpredictable, so you really cannot understand what would be the impact of changing environment on that system. The changes are generally non-linear, so the amount of impact or perturbation that you're putting on the system need not have a similar impact on the function. They are generally open systems, so they do interact with the environment. They are robust, so they have a certain range in which they can operate. They are modular, and this concept is very important, because if you've heard of this old fable of Hora and Tempest, where they were both watchmakers. So one of them both of them had the same principles for making watches, but one of them was very successful and made a lot of profits while the other one didn't. And that's because the one who wasn't successful would put together one part at a time and build up his watches, and if he couldn't finish it by the end of the day, he would just keep the watch down and it would break apart. Whereas Tempest, who was the most successful one, would build the watches as parts of 10. So he would build modules and then he would put them together. And so even if he was unable to build the whole watch by the end of the day, he would be able to, he wouldn't lose out, he wouldn't have to start all over again the next day. So modularity is key in complex systems. They also are self-organized and cooperative, and there's always a hierarchy that exists within a system. And so essentially with all these properties, systems essentially are more than sum of its parts, and they exhibit complex behaviors of adaptivity, of dynamicity, of goal seeking for a particular function, and they can even evolve. So there's natural selection that happens within them. So all this brings us to the concept of systems theory, which essentially talks about the behavior of a system and how it depends on the properties of each component in a system and the interactions between these components, and which is dictated by the environment. So therefore, essentially, in systems theory, there's an input, there's an output, there's a process that sort of connects the two, and there's always feedback. So this feedback is one of the reasons that leads to the non-linearity property that I talked about a little while ago. And essentially, this is the whole concept of systems thinking or systems theory is one needs to forget about reductionism and think more holistically. And this word comes from the Greek word holos, which means the whole, the entire, or the complete. And as the theory suggests, it is a very multidisciplinary field, and you need to evoke principles from multiple domains of study to be able to apply systems theory to any system. So let's move on to the concept of integrative biology and systems thinking in integrative biology, which essentially was a paradigm shift in the last 2 decades or so. So essentially, the first part of it is that you break down a cell into parts to understand life. And with the advent of a lot of machinery, measurement devices, the cell has been broken down to understand, populations of cells, individual cells, and all the molecular components that exist within a cell. And this approach, which was predominant in the previous century, is called the reductionist approach. So if you remember how I defined the system, you need to understand the elements or the components of the system. So in biology, in a biological cell, these are the components of the system. And then you need to, in the systems thinking, you need to look at the cell not as these individual parts, but how they come together. So you need to integrate the parts to understand the function, and that understanding can come through looking at crosstalk. So you basically integrate from, say, the genome all the way to pathways, and from pathways to understanding how mechanisms come up within the organism. And this is called the integrative approach. And for both of these approaches, data is very critical, and you all know that this is the era of data, so I will not harp on that. So one needs to look at what is the intersection of data that comes from all this experimentation on the left, and how one can understand the interconnection between each of these data types. And together at the intersection is what falls the concept of systems biology, which is basically applying systems theory to any biological system. So how many in the audience have an understanding of biology? A few. Okay, all right. So I'll start with one very elementary concept that I think we should have learnt in high school, which is called the central dogma of biology. And that, along with 5 ideas that have been built up over the century, they form the pivot between systems thinking and practical applications in biology. So the central dogma of biology is that you go from DNA to RNA to protein. DNA is is the genome or the genes. They convert into an active form, which is called the RNA. And this RNA actually changes depending on the environment that you put this DNA in. And this RNA can then be translated into what are called proteins, and proteins are generally the machinery that can help you convert one chemical to the other, that can help you regulate how much of one chemical goes to the other. It can help control, it can be a control element like a process controller to be able to control a third process. So this is the central dogma of molecular biology, and essentially the process, DNA is self-replicating. So the process of replication is basically you convert one piece of DNA or one strand of DNA into multiple strands. Then you can transcribe, then this transcription creates information. That information is in the form of a single strand, and when you translate that single strand using certain rules, a code of life, you can get proteins. And in the body, or inside any cell, it's not a monogenic relationship, it's not a one-to-one correspondence, but there are many. We all know that the human genome is more than 20,000 base pairs, and more than 20,000 genes. It's about 3 trillion base pairs. And I think if you run a string all the way from Earth to the moon, that's the number of base pairs that the human genome has. E. coli, which is an organism that you've probably heard about and is common, is probably on this surface as well, has about 4 megabases of 4 megabases that comprise its genome. So what are the 5 great ideas in biology that sort of form the pivot between systems thinking and applications of biology? So the first one is that the cell is the basic structural and functional unit of life. And that is why a lot of research, if you look at research done across the world in diseases, it is about the cell. It is cell biology that is dominant. The next important concept is that gene is the basis of heredity. And as I mentioned in the molecular dogma, the DNA replicates itself, and when you go from one cell to the other, one cell becomes two cells, and one DNA essentially becomes two bits of DNA, and it goes to the two daughter cells. So that's how you propagate, and that's how DNA moves from the parent to the daughter cells, and from the daughter cells to more daughter cells, and so on and so forth. So this is the process of heredity, and within this gene replication or DNA replication exists a lot of other smaller processes like regulation, control, and so on and so forth. Uh, the third important idea in biology is that evolution by natural selection is a characteristic of life. And I'm sure that all of you have heard about Charles Darwin and his Origin of Species. And he basically came up with two main tenets of evolution. One is that the main point in the theory was that there would be survival of the fittest, and so if there's a competition, there would be one that would be the winner, and that winner is decided based on what changes or how adaptive that particular species is, and that is attained by mutation and by selection. So there were two properties to survival of the fittest. One was mutation and the other was natural selection. The fourth important idea in biology is that life is based on chemistry, and this comes from the fact that most of the life processes that take place happen due to metabolism. So metabolism is the key chemical engine of life of a cell, and this chemistry dictates what are the proportion of nutrients one has to take, how does one nutrient convert to the other, how do you even create these DNA, RNA and protein that form the central dogma of molecular biology. Because in the end, DNA is a combination of 4 base pairs: adenine, thymine, cytosine and guanine, and all of these are nucleotides or nucleosides. So is RNA, and so are proteins. Proteins are made up of amino acids, as you probably know, and a bunch of amino acids come together in a specific manner to form a protein. So chemistry is sort of the key engine of life and life processes. The last but not the least, biological organization based on logical and informational processes and structures. This forms the fifth key idea, which essentially transcends into the idea of systems biology or systems thinking in biology. So essentially, this means you move beyond focus of the individual DNA or the individual RNA or the individual protein as prescribed by the central dogma of biology, and move to looking at systems as a whole. And so if you were to scale from this DNA, RNA to protein dogma to systems thinking, it would go from DNA to gene, gene to genome. So the concept of scaling holistically for every organism in terms of semantics is just adding an "ome" at the end of the word. So DNA, a lot of base pairs of the DNA form a gene, and if you look at multiple genes in an organism, it's called a genome. The RNA, which is basically transcribed from the gene, when they all come together, that's called the transcriptome, because there's a single transcript or a single strand, which is called the transcript, that is expressed from the DNA. And when you look at the whole of all the proteins that come together inside a cell, that's called the proteome. Now, where do the chemicals come in, right? So there are a set of proteins that convert one chemical to another chemical. So if the chemical is, say, you all have had a cup of coffee or tea this morning, so you would have consumed some sugar of sorts in it. So if you look at sugar, sugar consists of glucose and fructose. So glucose and fructose are actually similar in molecular composition, they're just slightly different in structure, and so they are isomers of each other. So these proteins can catalyze reactions like this isomerization reaction between glucose and fructose. So that is the last part, which is not traditionally included in the central dogma of biology, which also forms another aspect of systems thinking, another molecular hierarchy, if you will, inside the cell, and that connects to the fourth point, that life is based on chemistry. So basically you have a genome, you have a transcriptome, you have a proteome, and then you have a metabolome, which looks at the cell holistically from a molecular point of view. Now, when there's a crosstalk between each of these ohms or each of these molecular hierarchies, they give rise to more complex function. They give rise to control, they give rise to feedback, and all the interesting properties we know that exist in a complex system. So essentially these components, so essentially the function is more than the sum of its parts, and so the whole is more than sum of its parts. And since some of the work that we do in the lab focuses on cancer, I'd like to bring your attention to this book, The Emperor of All Maladies, by the eminent Pulitzer Prize winning clinician Siddhartha Mukherjee. So one of his quotes in this book is, "How can one capture genes that behave like ghosts, influencing cells from behind some dark curtain?" So in my opinion, this quote actually refers to the fact that although cancer is indeed well known as a genetic disease, one cannot really understand the outcomes of this disease because of the different molecular hierarchies that exist in the cell and the upstream and downstream factors that impact these genes. Another quote in this book emphasizes how cancer exploits the fundamental logic of evolution like any other illness. So he brings out this very scary question that if we as a species are the ultimate product of Darwinian selection, then too, so is also this disease which lurks inside us. So this itself explains the complexity of cancer in a way that one needs to understand crosstalk between all these molecular hierarchies, all the way from the DNA to the mRNA to the proteins and the metabolites, understand their interaction, which can also be, fancifully coined by a term called the interactome, to understand their interaction, to eventually result in understanding why cancer is such a complex disease. So some of the work that we do in our lab, the systems level work that we do in the lab, essentially combines technology with computation to understand biological insights. So we use a lot of different technologies. Like I mentioned, we try to probe the central dogma of molecular biology, but more on a holistic scale. So we look at the genome, or the exome. Exome is the coding part of the genome. There's a lot of junk base pairs in our genome. So we don't look at those junk base pairs, not because that they're truly junk, but we've not yet understood the function of this part of the genome. So we look at the exome, we look at transcript abundance or the transcriptome, we look at metabolites inside the cell. We look at respiration and growth. Respiration and growth are complementary in some cases, but at the cellular level, they can be distinct. There could be a cell that can grow on a particular nutrient like glucose and also respire on it, but there could also be another amino acid like serine, where it would respire on that amino acid, but it would not grow. So respiration is essentially the formation of energy and reducing equivalence, whereas growth is increase in the cell number. So you actually can dissect these two very nicely in the lab. So we use these kind of technologies. We use a lot of computational methods, both data-driven as well as mechanistic modeling, to understand, to analyze this data that we get from these technologies, and also to integrate, to create a platform to integrate this data. And then some of the mechanisms in biology, or the insights in biology we want to understand, are mechanisms of resistance, drug resistance and sensitivity. How do we predict chemosensitivity? So how can you identify new drug molecules that already probably are known to exist? Can you predict that these cells that are resistant to the primary line of treatment, will they succumb to these particular drugs, and then come up with an integrated paradigm for resistance. So this whole thing is basically systems thinking or systems biology of cancer, and we focus on a cancer called glioblastoma, which is the cancer of the brain. I'll come to that in a few minutes. So what exactly is cancer? So all of you have heard of cancer, right? So what exactly it is that cancer begins when one single cell, just one single cell, begins to grow out of control. But then I mentioned earlier that the cell, right, those 5 great ideas in biology. So within that one cell, there's the DNA, which is the hereditary marker. There's a lot of chemicals in there, all the metabolites that go on. There are transcripts that are formed, and there's an information flow. So this itself shows you how complex a system that cell is. So it's rather ironical that we say that cancer begins when only a single cell actually begins to grow out of control. So there are many hallmarks of cancer that have been deduced so far, and this is a figure that shows that. And since we are looking at the impact of metabolism or the chemical space on these in these cancer cells, here I've depicted that outside in this white space, this is all the metabolic space, and those 5 or 6 different markers over there, the Y, the plus, the infinitive proliferation potential, etc. These are the main hallmarks. So this figure essentially shows how the metabolic space impacts the hallmarks of cancer, and those are the arrows that point outwards, and the ones that point inwards are the ones that show how metabolism contributes to these hallmarks. So the chemistry, so as I already mentioned earlier, cancer is a genetic disease. It's known to be a genetic disease. It starts when a single cell begins to grow out of control. And there are factors both in the chemical space, the metabolic space, the gene space, the DNA space, and the flow of information between the two that is enormous, that is huge, that is very complex, that creates the emergent properties of cancer. So essentially, cancer is a multi-hit and a multifactorial disease. By multifactorial, I mean that there are many factors that can actually impact it. That could be the environment, the nutrient availability, the pH, so how acidic or basic is the cell inside, the oxygen, how much oxygen is available to the cell. And it's a multi-hit disease, as in it can hit different parts or different aspects of a cell, which are basically the DNA, so the genome can change, the transcriptome, the RNA can change, the proteins can change, and the metabolites can change. And all this impacts the physiology of the cell, which would eventually impact the outcome of cancer. Another important aspect is that the chemotherapy that is given to a cell, that can also impact, it's both, it forms a hit as well as a factor. So it can, it's a factor that impacts how the disease progresses, and it also eventually results in metastasis, and it can also cause resistance through being unable to kill the cancer cell. And then eventually after metastasis, you have recurrence once again, once the disease is cured. So it's a really, so this slide essentially just talks about how complex and a multifactorial and multi-hit disease cancer is. And it's further complicated through the ideas of metastasis and recurrence. So glioblastoma is a brain tumor, and it starts from cells in the brain. It generally demonstrates infiltrative growth, so that basically means you're mixing black and white sand together or salt and pepper together. So that is why it makes it very hard for a surgeon to surgically incise a brain tumor. The incidence is highest in patients who are in the prime of life, that is 45 to 55 years old, and the median survival is only 15 months with the best current therapies. Some of the hallmarks of the tumor, in addition to necrosis, that is the dying of tissue, microvascular proliferation, is also increased metabolic demand. So the prognosis, as I just mentioned, is very poor for glioblastoma. This treatment that exists are surgery, radiation, chemotherapy. There has been, so Temodar is a chemotherapeutic agent that came up in 2002, and within a year, resistance was shown to this particular drug. Although Temodar did improve the survival rate of glioblastoma patients along with radiation therapy. So there is a need for new treatment options, or a need to understand why glioblastoma cells get resistant to this drug and how do you resensitize them or identify alternate treatment strategies. So the model system we use in the lab is a cell line called U87, and what we observed when we grew these cells is that they had two different morphologies. So generally, mammalian cells, the cells that are inside our body, are adherent. They go, if you grow them in a flask, they would go and attach to the cell surface. But what we observed is that a part of them did go and attach to the cell surface, and some of them floated on top. Generally, for a cell biologist, this is bad news, because cells that float on the top mean cells are dead. But there was a very talented student who was doing this, and we weren't willing to believe that these cells that floated to the top were indeed dead. So what we did was, we did a very simple test. We collected all those cells and we did a dead or alive test. It's a chemical that we put in called trypan blue, and you can understand whether cells are alive or dead. And we saw that these cells were indeed alive and they were not dead. So the next question we had is, hey, if they float up to the top, are these the ones that eventually metastasize? Are these the ones that sort of evade the primary line of treatment, and so on and so forth? So the first step that we followed was, we separated these two cells, not just by filtering them out, by using some sophisticated techniques called fluorescence assisted cell sorting, and we looked at how do these cells grow. So the curve on the left, the blue one, essentially shows the parental mixed population that grew at a doubling time of 36 hours. That is, one cell doubles in 36 hours to two cells. And then if you looked at the separated cells, the spheres, which are called the neurospheres, and the adherent cells, which are called U87Mg, they both seem to have a different doubling time or a different growth rate. So the neurospheres, the cells that floated to the top, they seem to have a lower doubling time, so they took longer to divide than the U87, which took a slightly lower time. Then we decided to probe whether how these cells responded to the primary drug that was used for treatment. And so we used temozolomide, which is the drug that is used for glioblastoma. And then we saw that there was a difference in the IC50 value, that is the amount of drug that is required to kill 50% of the cells. And the change was almost 40%. So then we understood that this population required was either drug resistant or it was drug tolerant, because these two are two terminologies that are slightly different. Then the next question we had is, hey, these are spheres, they don't really form the nice star-like morphology that a neuronal cancer should form. So can we differentiate these cells? Can we add certain factors? Are these cancer stem cells, and will they again redifferentiate into the star-like morphology of the U87? And then we did those series of experiments, and within 4 days, we saw that these circular cells that floated on top, that initially we thought was an artifact of the way we were culturing cells and were dying, actually were cancer stem cells with all the markers that define a stem cell, and were able to differentiate within 4 days back into a brain cancer cell. So once this was done, once this was done, what we wanted to understand is, how do we now resensitize this cell that needs 40% more drug to that same drug? Would that be possible? Or can we identify other drug molecules that can, target these resistant cells? So as I mentioned earlier, there are each cell contains, basically each cell is programmed to execute the central dogma of molecular biology. So we decided to look at how these molecular hierarchies in the central dogma, how have these changed between these two types of cells. So we basically did what I showed earlier in that picture. We profiled for understanding the genome, the transcriptome, the metabolome, and the proteome. And you can catalog all these changes. And in order to study the interaction between them, we use what are called as constraints-based models. So we created these models and we used these heterogeneous data types, like I just mentioned, as constraints in these models, and were able to compute the cell phenotype. So essentially I have two cells now, one that is sensitive to the drug, the other that is resistant to the drug. I have profiled it to understand all the molecular components in each of this. I've done it in a very holistic manner. Now, just by knowing what are the components, I don't really know why this particular cell requires 40% more drug than the other. So I put them into a model and I predict the growth of the cell and then understand what is the redistribution that has happened in the pathways inside the cell that cause this increase in the need for the drug. So on doing this, so there are many methods that we've developed, and these are in a toolbox. And once you have these, you can probe what is the space. So I'm giving an example of only the, so one thing to note here is that this exome over here, this is the genetic space that we are looking at. The respirome is sort of a functional space that we are looking at. We are looking at how different nutrients change the way, the patterns of respiration between the cell that is sensitive to the drug and the cell that is resistant to the drug. And the metabolome is sort of the chemical space that exists within the cell. So we put all this in the context of a model, and these are genome scale models. So these are basically holistic models again. They have information, they are mechanistic models, and they have information of how you move from one gene to one protein to one metabolite, all encoded within the model. And so you are able to combine the biochemistry, the genetics using mathematics, and you are able to predict or compute cell phenotype. So one of the most important phenotypes in a cancer cell is growth. So we essentially use growth as a constraint because there's a difference between the two cells in terms of growth. There's a difference in terms of the gene space, the chemical space, and the functional space. And then we try to understand, is there any rewiring that has happened inside the cell? So these are some of the pathways. You really don't need to understand what they are at this point. But what I just want to show is that we saw, based on these histograms, that there was a difference in the way each of these cell types behave. The red part of the histogram essentially shows the cell that is resistant to the drug, and the blue one is represents the cell line that is sensitive to the drug. And as you can see, the area under the curve, the position, the height, everything is very different. So there has been a lot of reprogramming that has taken place in very critical pathways in the chemical space between these two cells. And then essentially based on this reprogramming, we identified drugs that can target those rewired pathways. And then I'm just showing an example, a single example of that. So rotenone is one such drug that impacts respiration. And if you look at the cell line that is sensitive to the primary line of treatment, it requires much higher levels. It requires 1.8 micromolar levels of the drug of rotenone, whereas the neurospheres, which are the resistant cell lines, it requires only 5 nanomolar. So there's a 3 order of magnitude difference between the levels of rotenone that are required to kill these cells. And there were also another set of 14 other drugs that we found that were different, based on, had different response based on the rewiring. So this is one way one can use the systems approach to probe cancer cells to understand the phenomena of resistance, which is an emergent phenomena. So another important problem is the problem of infectious disease, and more than 20% of the deaths in the world are because of infectious disease, and even within the non-infectious disease deaths, around 20% are related to some infection that is contracted by the patient in the hospital. The drug pipeline over the last 36 years, there have been no new antibiotics that have been approved by the FDA so far. And drugs are becoming less effective, as I mentioned earlier. The bugs are becoming smarter and they're able to evade, the impact of the drug. And there's always a complicated profit motive based on the business models that currently exist in the pharma. So there's not much incentive for the pharmaceutical industry to make more antibiotics. So this is an article from 2008, and essentially it talks about the success of genomics. That is basically for every bacteria or pathogen that exists, every bug that causes disease. If you look at the genome of that drug, initially it was thought of being the holy grail, that you would be able to sort of understand why bacteria become resistant to these drugs, and how would you get beyond that, and how would you design new molecules. So that is what this the commentator from GlaxoSmithKline says, that to his surprise, although more than 650 bacterial genomes have been sequenced, it's been a dismal failure, and it's not yet yielded the treasure trove of new targets that they had once thought it would. So, and they conclude that genomics is simply not a good paradigm for discovering new antibiotics. But what I feel, the argument that my school of thought comes from, that it's not that genomics is not a good paradigm, but there is a need for systems thinking here, where you need to understand the crosstalk between these genes with the transcripts, with the proteins, and the eventual chemical engine, because resistance is essentially growth which happens because of this chemical engine in the presence of the drug. So you need to understand each of these molecular hierarchies and understand the crosstalk between them to be able to use genomics as a very good paradigm. And I'll quickly show one example here, where we took a zoonotic pathogen, that means this pathogen causes disease only in animals, and we made them resistant to these two antibiotics, streptomycin and chloramphenicol. And they both impact a part of the protein synthesis machinery. And essentially we did what we do best in the lab, that is we profiled the genome, we looked at some phenotypic characteristics, what are the outcomes, how do these bacteria grow under different conditions, and so on and so forth. We looked at the metabolome that I just discussed, and then we put them in the context of a model, the genome scale model that I talked about. And what we understood was, by profiling each of these molecular hierarchies, we could understand what was happening within the cell, but we still couldn't understand why a certain cell was resistant to that antibiotic. But by putting it into the model and by predicting phenotype, we were able to understand that there were these certain ratios that were going up and down, which basically control redox potential inside the cell. And then when we identified compounds that can target this redox potential, we saw that the cell died. So we could resensitize, we could identify a predictive mechanism for killing, and we could resensitize these bugs that were resistant to the antibiotic back once again. So I'll just move on in interest of time to how you can use systems thinking for global problems, or is it really a good paradigm for global problems, right? So there are a lot of systems that exist in nature. There are ecological systems, industrial systems, and social systems. You can use systems thinking for each one of them. As we know, a lot of the natural systems that support Earth have been disrupted so far, and that's why we have global warming and climate change that has, plagued us all. Industrially, the industrial system is very useful to us because that's where the bioeconomy lies. That is where we can make products. We can stop using petroleum-based products, and we can help, we can help make things more green, which will eventually again impact the environment. And of course, the social problems, the social system is an intangible system that's created by humans, and you can also use systems thinking for, say, policy decisions and so on and so forth, but we won't go into that today. And essentially the whole of the last few centuries has been about the reductionist approach, and therefore it is critical for us to look at this concept of thinking in systems to solve these problems today. As Einstein said, the problems we've created in the world today will not be solved by the level of thinking that created them. So we need to move beyond the reductionist approach. So how, as a biologist, right, how can you apply the understanding in biology? Because biological systems are far more complex than any system that exists on planet Earth. How can you use the understanding in biology or the methods in biology to other fields? So this is very well captured in this small commentary, Can a Biologist Fix a Radio? Where essentially this particular biologist tries to bring in concepts of systems thinking and says that if there's a broken radio, there are a few steps from the systems thinking that you would use. That is, you would embrace more of a black box mentality, look at the input and the output and look at what has changed. You would systematically perturb each component in that circuit and observe what was happening, understand the function of each of the components. You would then create a network map to understand how these different components interacted with each other, and was there any connection between the components that was broken. You would also do some measurements to understand, hey, whether the sound was going high or low, some quantitative measurements, and this would be a model guided diagnosis and repair for this process to fix a radio. And essentially in the end, you would be able to differentiate between a serendipitously recovered component, a really important component, versus the most important component, which would actually tell you how to fix your radio. So coming to different areas, if you look at finance, right, in 1929 there was a Wall Street crash, and this is a stark reminder of the complex dynamics in financial systems and the importance of understanding these systems from a holistic perspective. The behavior of the whole system cannot be predicted by just understanding the behavior of individual components. So we all know that the stock market crashed, but some of the most unexpected and unforeseen consequences, which are the emergent properties, were the rise in suicides, political instability, and social unrest. So these are some of the properties you would not have predicted by just looking at the properties of the individual components that are a part of the financial network. So essentially, we need to use systems thinking to solve a lot of problems across different fields. We need to look at the big picture. We need to examine how things relate. We need to take a wider perspective by peeling back the layers of the onion, look for root causes and improvements, and challenge and change our paradigms. So essentially, emergence-led biology isn't just a scientific trend, it's a call to action. And the symphony of emergence is all around us. It's waiting to be understood, harnessed, and celebrated. And before I end, I'll leave you with a picture of my lab. Thank you, and I'll be happy to take any questions. Thanks for your attention.